Hello everyone. So in this video we will discuss about biotechnology principles and processes. This is unit 9 of class 12 biology. So from this part almost 11 questions asked every year, almost, and this chapter contains a major weightage, so you should prepare it thoroughly. It will definitely help you in increasing your marks in Neet 2024. Definitely there are some syllabus changes has been announced for the next examination 2024, but this chapter, this unit will definitely be asked. So preparation of Pyqs and concept thoroughly will really help you in your ranking, in your marks. And I will upload all the Pyqs, which is more than 200, from this unit 9 with some important concepts and tips and tricks. Okay, so you can take help from this video. So let's start with the questions. In this video we will discuss about principles of biotech and tools of recombinant DNA technology. Let's start with the questions. Here comes the first. The DNA molecule to which the gene of interest is integrated for cloning is called. Answer is vector. The cutting of DNA at specific location became possible with the discovery of which enzyme? Restriction enzyme. Which of the following is a case of wrong matching? Vector, site of tRNA synthesis, callus, an unorganized mass of cells produced in tissue culture, micropropagation, in vitro production of plants in large numbers, and last, somatic, fusion of two hybridization diverse cells. So the answer will be vector DNA, because vector DNA is not a site of tRNA synthesis but carry a foreign DNA segment and replicate inside the host cell. They are used in recombinant DNA technology. Okay. Next question is, which of the following techniques made it possible to genetically engineer living organisms? So this is recombinant DNA technology. Fifth, which of the following is used in gene cloning? Plasmids. Sixth, the bacteria generally used for genetic engineering is Agrobacterium. Okay, so this part will be asked from tools of recombinant DNA technology. And here's the first question. Which of the following is not true regarding gel electrophoresis? Okay, so the answer will be A. The presence of chromogenic substrate gives blue color DNA bands on the gel. This is not the correct about gel electrophoresis. The presence, while the correct answer will be the presence of chromogenic substrate gives blue colored DNA bands on the gel. It's not correct. Is the incorrect statement, as bright colored bands of DNA can be observed in the gel when ethidium bromide treated is exposed to UV light. Okay, so it always asks, when do you see DNA on the gel? When you put this dye in agarose gel, then only you will get visualize the DNA. Okay, so the next question is, in the following palindrome base sequence of DNA, which of the following can be cut easily by particular restriction enzyme? Okay, so here the answer will be C, because this is a palindromic sequence. Palindromic sequence is a DNA sequence of base pair that reads the same on the two strands when orientation of reading is kept the same, like from 5' to 3', then 3' to 5'. From here to here the orientation will be the same. So option C is the only palindromic sequence. So this is the correct. Restriction enzyme cut only the palindromic sites. Next, which of the following is not a desirable feature of cloning vector? Answer is D. Presence of two or more recognition sites. Because the feature of desirable in a cloning vector are presence of origin of replication, presence of marker genes, presence of very few, or preferably single recognition sites, not more than 2. Okay, so this is the correct answer. This is not desirable feature of cloning vector. Now, fourth question is, given below are two statements. Statement 1, restriction endonuclease recognizes a specific sequence to cut DNA known as palindromic nucleotide sequence. And statement 2 is, restriction endonuclease cut the DNA strand a little away from the center of the palindromic sites. So which of the following option is correct? So the answer is both statements are correct. Okay. Next, DNA strands on a gel stained with ethidium bromide, when viewed under UV radiation, it appear as, so the answer will be bright orange bands. Okay. Now, plasmid PBr322, which is very common in NCERT, mentioned it, has PST1 restriction enzyme. Okay, this is plasmid, this is restriction enzyme within gene ampicillin resistance that confirm within gene AMPR that confirms ampicillin resistance. If this enzyme is used for inserting a gene for beta galactoside production and the recombinant plasmid is inserted in an E. coli strain, what will happen from this option? So the answer will be, it will not be able to confer ampicillin resistance to the cell. Why? So in plasmid vector PBr322, two unique restriction site present, PST1 and PV1, are located within the AMPR gene, and BAMH1, SL2, etc. are located within the tetracycline gene. These all are plasmid instruction in NCERT, and if you want me to make a video on this, you should comment and I'll make a video on it if you want. Okay, so now the presence of restriction site within the marker genes tetracycline and ampicillin R genes permits an easy selection for cells transformed. When restriction enzyme BAMH1 or SL1 is used, the DNA insert is placed within the gene TET-R, making it non-functional. If this enzyme is used for inserting a gene for beta galactoside production and the recombinant plasmid is inserted in an E. coli strain, it will not able to confer ampicillin resistance to the host cell. I hope you have understood this. So move to the next question. A specific recognition sequence is identified by endonuclease to make cuts at specific position within the DNA. Is which? So restriction enzyme cuts always within the palindrome nucleotide site. Next is, identify the wrong statement regarding restriction enzymes. So the answer here will be B. Sticky ends can be joined by using DNA ligases. This is not the correct. 9th, choose the correct pair from the following. Okay, so answer will go clearly to ligase. Ligases are the enzymes which joins two DNA molecule. Okay, next, the specific palindromic sequence which is recognized by EcoR1 is what? So the answer will be this. This is written in NCERT. This is the specific sequence which is recognized by EcoR1. Okay, so if you like this video, please like and subscribe for further all the classes and PYQs, and be with me. I will cover all PYQs of biology class 11 and 12 for Neet 2024. Okay, so